Good afternoon, everybody. Welcome to La Jolla Institute for Immunology. I'm Dr. Erica Ullman-Sapphire, the President and CEO at LJI, and I am thrilled to have you all join us here today for our Life Without Disease lecture and reception. The record-breaking number of registrations for this event, along with your collective presence here today, reflect a powerful shared dedication, advancing our understanding of the immune system and driving scientific progress forward to a life without disease. For those unfamiliar with LJI, allow me to share a little bit about who we are and what drives us. For over 35 years, LJI has led the charge in tackling some of the most formidable health challenges by maintaining a steadfast focus, unraveling the mysteries of the immune system to better combat disease, ranging from cancer to viruses to autoimmune disorders. The Institute is a hub where rigorous scientific inquiry merges with innovation and collaboration, fueled by a diverse team of brilliant minds who push the boundaries of conventional research. Our work is made possible by the exceptional leadership and generosity of the Institute's highly accomplished board of directors. Their visionary guidance and their philanthropic support are instrumental in propelling our mission forward towards life without disease. We are deeply grateful to have several directors with us here this afternoon. Now, today's lecture, Outsmarting Autoimmune Disease, is made possible thanks to the generous support of the Global Autoimmune Institute, or GAI. For those who maybe not be familiar, GAI is an organization that goes beyond traditional advocacy alone by integrating research, awareness, and community support into every aspect of their mission. GAI is dedicated to transforming the landscape of autoimmune disease diagnosis, treatment, and patient support. The organization funds scientific research, develops comprehensive educational resources, and champions a multidisciplinary approach to healthcare. And with a strong emphasis on community engagement and awareness, GAI empowers patients, their families, and healthcare providers by bridging gaps in understanding and addressing the complexities of autoimmune conditions. Their commitment to driving forward disease solutions has truly been transformative, marked by an investment of over $1 million in LJI just this year. And we are honored to welcome members of their leadership team who have traveled from the East Coast to join us for this discussion today. Thank you so much to GAI. And now it is my pleasure to introduce our presenter, Dr. Sam Myers. As LJI's Global Autoimmune Institute Assistant Professor, Sam is at the forefront of expanding our understanding of cellular decision-making. His lab is dedicated to decoding how immune cells process complex information. He leverages advanced proteomics and mass spectrometry to uncover the biochemical signals that determine cell fate, how one cell turns into another and develops. These investigations reveal how disruptions in those intricate pathways can lead to autoimmune, inflammatory, and other diseases. And so here's a little bit about Sam himself. He earned his Bachelor of Science in Biochemistry from California Polytechnic State University. And he went on to his Ph.D. in Chemistry and Chemical Biology from the University of California, San Francisco, UCSF. From there, he did postdoctoral research at the Broad Institute of MIT and Harvard, where he sharpened his vision for integrating genomics and proteomics, setting the stage for his disease research today. We were very fortunate to welcome him as an assistant professor in our Center for Autoimmunity and Inflammation in 2021. In today's discussion, Sam will navigate us through the maze of how immune cells make complex decisions. Decisions that, when they go awry, can tip the balance from healthy immune responses to chronic inflammation and autoimmunity. He will show how his lab uses cutting-edge tools to map these molecular conversations and what this means for therapies that could modulate the immune response more precisely than ever before. After Sam's lecture, we'll move into a Q&A session where I will join him on stage, and that discussion will be moderated by Sandy Boak-Werness, an LGI board member and the founder and executive director of the Global Autoimmune Institute. Sandy brings invaluable insights as both a leader and an advocate for patients navigating these complex challenges, enriching the conversation and bridging scientific advancements with deeply personal connections. Sam will formally introduce Sandy following his presentation. And then following the Q&A, we invite you to join us for a gluten-free reception in the atrium. Invite that person too. Our reception will give you a chance to connect further with Dr. Myers and also to engage with the next generation of autoimmune and inflammatory disease researchers. So among them are Remy Morocco, Amparo Martinez Perez, and Nirmalo Dasgupta, each early career researchers here at LGI who bring fresh perspectives to the field as finalists in the 2025 cohort of LJI's Tully and Ricky Family's Spark Awards for Innovations in Immunology. Our young scientists are eager to discuss their projects and their work with you and to share what they are working on as they compete for prestigious Spark Awards. Also, Lizzie Dorner, who leads our mentorship program, cultivating the next generation of immunologists with opportunities for high school students and undergraduates. She will be here. That mentorship program is especially near and dear to Dr. Sam Myers, and that made it a special focus of this event too. So I encourage you to introduce ourselves to each of LGI's rising stars and learn more about their research. And especially as we are stepping into the season of giving and gratitude, I want to highlight that both that Spark program and our internship opportunities owe their existence to philanthropic support. Many of you in the room have contributed to these programs, and for that we offer you our heartfelt thanks. When we look at the trajectory of those careers that are launched here with those programs, and the origins of LGI's most significant scientific discoveries, so much of that can be traced directly back to your generosity and support. The impact is profound. Those findings are because of you, and we hope that you take pride in all that you have created and galvanized here. If today's discussion inspires you and you'd like to get more involved with LJI, our advancement team, including our Vice President for Advancement, Kelsey Dale, and all the rest of her group, are here to share opportunities to support and engage with our work. Now, let us welcome Sam Myers as he leads us into a world where science unravels the language of immune cells and opens doors to outsmarting autoimmune disease. Dr. Myers.
Thank you very much for that introduction. That was quite lovely. Well, thank you everybody for coming today. I'm going to talk about some of the work we're doing in the lab, but it'll be more about the kind of inspiration of why we look at the things we do and how we approach them in what I think is a different way than kind of traditional science. So we have outsmarting autoimmunity with cellular decision making by studying cellular decision making. And so our immune system balances health and disease, okay? And it keeps this good balance between promoting inflammation when we need inflammation and resolving inflammation when that inflammation is no longer needed. So for example, if there's a bacterium or a virus, right, we want to promote inflammation by building up more of these cells that can attack the virus or attack the bacterium so that we can eventually just eliminate those pathogens, okay? And once those pathogens are eliminated, we need to resolve the inflammation, right? So we need to reduce the number of pro-inflammatory cells, increase the number of, or the activity of anti-inflammatory cells, and restore that balance back to a healthy state, okay? And so you might think, why would we need inflammation? Well, inflammation is important for a lot of things. So for wounds, for cancers, for infections, right? We want inflammation to occur to take care of these things. Now, the other kind of flip, this other side of the coin is you can have too little resolution of inflammation. And so what happens if you have too much anti-inflammatory responses, or then that allows the bacteria or the virus to proliferate even more, right? So again, we need this balance to kind of keep ourselves healthy and protected from disease. Now, this balance is out of tune in autoimmunity, okay? There's an increase in pro-inflammatory cells or pro-inflammatory activity, or there can also be a decrease in anti-inflammatory activity or anti-inflammatory cells. Right? And so again, this is kind of the underlying effect. We have too much inflammation and not enough dampening the inflammation in autoimmune diseases. And autoimmune diseases are a multi-pronged and complex system. So there's over 100 different types of autoimmune diseases that have been reported. They happen to a lot of people. 3 to 5% of the population is afflicted by these. The etiology, or where these things come from, is also multi-pronged. There can be genetic predisposition, so your genetics can help determine one way or the other. But there's also environmental effects, lifestyle effects, diet, and again, fighting infections. On the right here, I'm showing kind of the more prevalent ones, but you can see they affect basically every organ in our body, from the skin and psoriasis to our guts and the inflammatory bowel diseases, or type 1 diabetes, rheumatoid arthritis, right? And one of the things that LJI has taken a particular interest in when we think about autoimmunity and how we're going to fight autoimmunity is the female prevalence of it. So you can see I have 3 women there and one male figure, and that's because for some reason women are much more susceptible to autoimmune diseases than men. So what this bar graph is showing you is both the percentage of how much of this disease is subjected to on women. So you can see Sjogren's syndrome, lupus, SLE. These are all 95%. These occur in women, right? And even in the ones where men take the lead in diabetes, right? It's still pretty even, right? So for some, and so we have a center here that's kind of based on this. What are the sex-based differences of the immune system and ultimately autoimmune diseases? And so what my lab does is we study the decision-making process of inflammation, right? So how are these cells deciding whether they need to upregulate pro-inflammatory activities or increase the number of pro-inflammatory cells? Or how do these cells make the decision to turn on your anti-inflammatory programs, right? And so this is kind of the decision-making that's underlying this balance between health and disease in the immune system. And cellular decision-making actually happens at every stage of our life, right? So this is early embryonic development. So at one point, when you were a single fertilized egg, I just learned how to do this the other day. No, I didn't. Okay, nope. Oh, no. Okay, we'll just do it by mouth directions. When you were a single fertilized egg, that fertilized egg had to make the decision to divide into two cells, right? And then those 2 cells needed to make the decision to divide into 4 cells, and so on and so forth, right? And until you got to this point where you're a blastocyst, and this is kind of like what you think of as the embryo, right? And these cells in the embryo are what we call pluripotent. That means they can turn into any type of cell in the body, right? So how do they make that decision, right? That's one of the kind of the earliest questions we ask in embryogenesis, right? But at some point, each of the cell of the embryo makes some decision to become some lineage. So the endoderm, which is your digestive tract and your thyroid, or the ectoderm, which is your brain and your skin. At some point, these cells all make the decision to become one of these cell types. And once they get their act together, they can then start just growing, right? They've already done their differentiation, and they can just start growing into a child. Okay. And there's actually cellular decision-making happening right now in this room. I mean, not a pregnancy thing. I don't know what's that. That I don't know. But your hematopoietic stem cell system, or your bone marrow. Your bone marrow right now has hematopoietic stem cells in them that are, and you don't need to really see the details of this. All you need to see is that there's one cell that we're pointing to in the upper left there. And what that's doing is that's sensing cues and making the decision to either go up this track and turn into one of these top cells, or go down this graph and turn into one of these other cells, right? And then at some point, these cells in the bone marrow at the top there in kind of the beigeish color, right? These cells then have to decide to go into the blood, circulate through our body, and then take residence in the tissue that they want to protect, okay? And so these cellular decision making is kind of happening on long time scales, right? Hours to days, right? But there's cellular decision making that's happening within seconds, okay? So what I'm showing here is that big kind of oblong peanut looking thing. That's a white blood cell. All the black, bigger ones are red blood cells. And then that little black double dot at the top, that is a bacterium, okay? And so the job of this white blood cell, what it's to do is it's to sniff out and destroy this bacterium, right? Without affecting or without attacking the red blood cells. So you can see, as it's pushing through, it's just pushing the red blood cells out of the way. It's deciding, I'm not going to attack you. I don't need to attack you. What I need to do is I need to chase that bacterium, okay? And again, these are happening in the time scale of like seconds, right? And eventually it catches the bacteria, engulfs it, and destroys it, okay? Now, if we take this super high resolution view of that microscope, that video, right? This is where signal transduction comes into play. So signal transduction is basically the cell sensing something in its environment, internalizing that signal, and responding appropriately, right? So as this bacterium flees, it leaves behind it inflammatory molecules that our white blood cell has receptors for. And these inflammatory molecules will bind the receptors and transduce a signal into the cell, okay? And the way I'm showing it here is that there's one little concentration of these inflammatory molecules that's really high, and so you get a big signal from the high concentration of these molecules, right? We're down here in the bottom right, there's like a little bit of the signal, so you get a smaller signal transduction here, and then the receptor there at the top isn't sensing anything at all. And what this does is this tells the white blood cell, this is the direction I need to chase this bacterium in, right? So it's transducing that signal across the membrane and saying, well, I'm seeing more of it over here, so I'm going to go that way and not go that way, okay? So again, I've given you a couple examples of how cells make decisions and what I mean by cell decision making. So how do we study cellular decision making? How do we do it in the lab? Well, I'll take you back to the central dogma of biology. DNA in every cell of our body transcribes into mRNA, and that mRNA, that messenger RNA, is then translated into a protein, okay? And so the way I kind of think about this is that the DNA is, the information storage of all the possibilities that a cell can do, right? Everything that it can make, how those products it's going to make are going to come together and form a function, right? Everything's in every cell, right? Every cell of our body has the same information in it. But then there's some selection process that says, well, I'm a cell and I'm going to turn on a subset of genes that are important for chasing bacteria, okay? And so that's where the mRNA comes in. But then the mRNA gets translated into protein, and the protein is what's actually doing the function. It's scaffolding the cell, it's rearranging the membrane so it is chasing the bacterium, it's doing chemical reactions in the cell, it's processing information. So the protein is where a lot of the information is actually occurring. And so the analogy I like to use is, how was the wedding, okay? So you think about when you're planning a wedding, you have all your possibilities. You have a handful of dates you can pick from, the venue, what drinks you're going to have, music and food, and then you have a list of people that you've invited, right? This is all the possibilities that your wedding could turn into, okay? And then at some point you have to make decisions, right? You pick a date, you have a venue, maybe a backup venue, right? You send out your invites and you've gotten responses, and this is kind of what you planned the wedding to look like, right? But the turnout is what you're going to talk about, right? You're going to say, well, my red dot friend at the top there didn't show up, but my blue dot friend did with two of their friends, Some people brought baby. One guy brought a dog and all his friends, right? Nobody ate the pink, they all ate the black. So you see, so the reality of the situation can be very different from what the plan was or even what the selection of the plan was, right? And I translate that back to the central dogma of biology, right? We have the genome, the DNA, and all the storage possibilities, the mRNA, the things that the cell picks to do, and then the protein, which is actually what is going on in that cell at that particular moment. A slightly more elegant and probably accessible example is a caterpillar and a butterfly, right? The caterpillar and the butterfly both have the same genome, they have the same DNA in all of their cells, but they're expressing different genes that turn into proteins that make them the caterpillar or the actual butterfly itself, okay? Okay, and then it gets another level of complication, and this is where a lot of the work that we do is the post-translational modifications to proteins, okay? So I said that mRNA gets translated into proteins, and then there are post-translational modifications that occur to them. I use them, I refer to them as almost like chemical ornaments on proteins, okay? So here we can have a protein of interest, that's what POI stands for, on the left there with just an amino acid in it. That's just the way it looks normally, that's the way it was translated. And then what can happen is it can go down two different paths, and it can get a sugar molecule attached to it, or it can get down in the bottom here, a phosphate attached to it. And so these are like little chemical molecules that can get added and removed from proteins in a very fast fashion, right? And so you can have multiple states of one protein of interest, right? So even having one protein there means it could be in 4 different states, and those all could be doing different things, right? They could be doing chemistry faster, they could be localizing in the cell differently, they could be telling the cell to do this, do that. So each modification state of a protein is important to study so that we know what the functions are going on, okay? And again, we can return to our wedding analogy to kind of visualize this. So you have all your friends at this wedding, right, which are the dots, and then they are post-translationally modified with a cocktail or a cigar or a child, okay? And if you just looked at individual, you might not really know what's going on, but what happens is they are drawn together and they are grouped together in their function by the way they're post-translationally modified, right? So people with the baby and having a couple cocktails, they might go to bed really early, where the people having cocktails and smoking cigars might be up all night, okay? And you can infer what's going to happen and what the functions are by looking at these post-translational modifications, looking at these chemical ornaments on proteins, okay? And then so we typically draw signaling pathways like this, signal transduction pathways like this. So in the blue, we have a receptor, and that's that kind of green molecule that would like bind that little jalapeno, the little inflammatory molecule from before. And then it'll turn on these pink proteins that will have a cascade of post-translational modifications that's called, in this case, phosphorylation, right? That basically says this pathway is turning on, and we need to turn on the blue response gene, okay? And so again, we see this signal, and traditionally what we do is we have some reagents that detect these specific, so the arrows are, again, just kind of a proxy for the chemical ornament, right? We have some reagents that can detect that one specifically, and we can say, okay, we know those two are there, green check. We tested out the red check, and it wasn't there, so we're going to say the blue pathway is on, okay? In reality, it's a lot more complicated than that, okay? So these are called heat maps, and what we're looking at is that each row in a heat map is the equivalent of one of these arrows, right? So there's not 6 phosphorylation events going on in this pathway. There's hundreds to thousands that are dynamically changing over time, okay? And even in pathways that we assume are similar, the left and the right heat map, those are signaling through the same components, but somehow one is pro-inflammatory, one is anti-inflammatory, and you can see the differences, right? So even though we think they signal through the same 4 proteins, you can see there's thousands of things changing in the first 90 minutes of them sensing the molecule that they're sensing, okay? And then it's not just one gene that turns on. I showed that red and blue bar that said response A, response B. It's not just one gene, it's a whole block of genes that turn on, because that is the genetic program the cell needs to do its function. Chase the bacterium, attack the cancer cell, attack the cell in your knee. Okay? It turns on these programs to enact the function that it just sensed that it needs to do. And there's very little known about most of these chemical ornaments, okay? So this is, again, looking at just protein phosphorylation, where 5 years ago, there was about 250,000 of these reported in the literature. And we only have a function for about 3% of those. Now, I would say in those 5 years, that 250,000 has probably doubled, and the 3% definitely hasn't, okay? So, and then the other kind of bias that, the reason we do the work that we do here is that most of the ones that are reported are typically reported in cancer and neurodegenerative diseases, right? Where you can see that autoimmune diseases, inflammatory diseases, and immunodeficiencies are much less represented, right? Not because they're less important in these systems, just because they haven't been studied nearly as much, okay? And so that's where we're kind of trying to come in and make a difference here, okay? And so how do we get the whole cellular picture? So we have the signaling part, right, sensing the signal. We have the gene expression, which is kind of the response, right? So I use this as like the thought process and the decision that's made, okay? And you have to put those together to get the whole picture where you see what I call signaling to transcription networks, okay? And so what my lab does is we develop tools and technologies to see the whole picture, to put both things together. So we apply interdisciplinary approaches to study autoimmune diseases. Okay, and so one of the models and one of the cell types that we look at, one of the immune cell types that we look at are helper T cells, okay? So I know you can see even less of what that graphic was, but that's your bone marrow from before. One of those cells that eventually a hematopoietic stem cell turns into is a CD4 T cell, okay? And a CD4 T cell also has a lot of complexity. It can see, they're also very important for balancing our immune system, right? We have some cells down here that promote inflammation, and some cells down here that we generally consider anti-inflammatory, okay? But again, we want them at the right balance so that we're not over-inflamed or not being inflamed enough when we need it, okay? And then what my lab does is we study how these signals, so this list of molecules here, how these signals actually tell this naive T cell, this naive CD4 cell who hasn't changed into anything yet. It says, oh, well, I see TGF beta on here on the right all the way, right? So I'm going to become a Treg cell, right? And so we study the signaling pathways and the genetic responses to these signaling events, okay? And again, we apply these tools that we develop these new tools, and then we apply them to situations like this to say, what if we want to have more regulatory T cells, or what if we want to have fewer or more pro-inflammatory cells, okay? And so kind of just to wrap up, what I'm going to do is I'm going to tell you kind of two little vignettes of things that are actually kind of early on, but I'm really excited about them, and they fit really well into this talk. And so this is, I'll give you a more concrete example, like no more wedding stuff. We'll get into it now. One of them is through a former research technician in the lab, Patrick Kennedy. He went to UCSF for graduate school, actually, the same place I went. And that's something that we've kind of worked on. And then the other project is very fledgling, and it's Maria Matias, who's a postdoc here, who's actually a former Spark winner herself. So that's kind of how this project got started. Okay, so vignette number one. So functional signaling in T cells. Okay, again, this is a similar idea, just kind of shaped differently. We have a T cell that's resting. We give it a shock. We activate that T cell itself. The signaling pathway propagates through the cell, and then it reaches these colored circles, which go and then turn on genes. And we'll say we have the green and blue genes, okay? Again, we built a new technology here to study the thousands of phosphorylation sites that are occurring in this. Typically, again, you study 5, 10 at a time. In this experiment, we were able to functionally characterize about 10,000 of these, okay? And so this is kind of the graphical abstract. This is kind of just an idea of what happens. So we do our measurements in the pink space, right? We look at all the signaling events that we can find. And then we use these new genome editing tools to then go in and make it so that phosphorylation event can't happen anymore, but just that one. We leave all the other 900 or 9,999 alone. We just mutate that one so it can't happen anymore. And then that's where we have the gray cells. They get mutated, so they're these colored cells, okay? We shock them, or we activate them in some particular way. And then we measure how much green are they making, how much of this green gene are they making. And if they make more of this green gene than all the other ones, then that gives us an idea. If they make more or less of this green gene, that means that mutation that we made, that phosphorylation site that we blocked, was important in some way or another, right? It had a function relative to making this green gene, okay? And I won't go through kind of the whole process because this is actually a paper that came out this year. But I'll show you kind of where we're going with this. So remember I said these genes turn on as, these transcription programs turn on as a group, right? A group of genes turn on. The T cell activation pathway is a group of genes, okay? What we're seeing here at the top is each row is a mutation that we made, and then they're colored based on the more light the color is, the higher the expression is. So again, like the top corner here, it's lowest in black, it's highest in white, and it's kind of middle in purple, okay? This is all thought to be a transcriptional program, but what we're seeing is we can break this transcriptional program up when we make that top mutation. That seems to increase those genes relative to the other mutations that we made, right? And we can start dividing these transcriptional programs up into smaller programs, right, just by making these really precise signaling mutations. And so why is this important? Like, who cares, right? Well, it turns out there are 4 genes in this T cell activation pathway that, using the database, the DICE database here done by Vijay's lab, right? We have 4 genes in here that are specifically expressed in T cells, only in activated T cells, and those expression levels are associated with autoimmune diseases, right? So the protein BOC2 doesn't really matter what it is for this, but it has a ton of associations with asthma, Crohn's, MS, right? All sorts of things, right? So the idea would be is that if we can start breaking up transcriptional programs, so it's not 50 genes turn on, 100 genes turn on, and we can modulate one gene, we could potentially start to ask, how can we tune the expression of BOC2, right? If we want it to go up or down to, provide some alleviation to these autoimmune diseases. So again, if we wanted to stay at the normal level, which is the yellow in this case, right, we wouldn't make any changes to the cell. The bottom one is kind of our control. We didn't make any changes to it, right? The top one, we can turn it down. We make that mutation. We can turn down BOC2. The middle one, we can turn it up, right? So as we do more and more of these, as we do more and more mutations, right, we can kind of focus in on BOC2 and say, what are the signaling events that we can look into in the pathway, reconstruct this pathway and say, where can we use existing drugs? Because most drugs are to signaling molecules anyway. Where can we use existing drugs to tune this so that we can tune the expression of BOC2 in T cells? Right. So this is something that we've kind of just started doing, and I've just convinced the graduate student it's going to work. And so, and he just passed his exam the other day. So 3 other professors actually think it's not a terrible idea. So, so that's one vignette. And then the other vignette is new functions for known cells. So I told you about CD4 cells, CD8 cell. It's a different flavor of T cells. What they typically do is they directly recognize a viral infected cell or a transformed cancer cell. And when they recognize it and the cell tells the T cell, I'm virally infected, the T cell will then release molecules to basically destroy those cells, right? They directly kill the viral infected cells so that the virus can't continue to make virus and spread more and make more, or it directly kills the T cell, okay? And I'll show you what this looks like. I found these videos. These aren't me. These are online, but you gonna do it? Okay. So the green is a T cell, and the blue is a cancer cell. And what it's doing is it's sampling the cancer cell, and by making this what we call an immune synapse, and then these lytic granules, these vesicles that have these digestive enzymes basically to chew up the cell, you can see that they're released towards the cancer cell itself, okay? And so again, now it's purple cells or T cells, and the big blue cell is cancer, and then you'll see these waves of, again, these lytic molecules that should be destroying the T cell. Okay, so you can see some T cells are coming around, checking it out, figuring out something's going wrong, and then release these molecules to destroy that cell, okay? And we want that to happen, right? We want that to happen to a viral cell. We want that to happen to a cancer cell. And that's what CD8 cells are known for. They're known for being able to directly kill. But they don't do a perfect job. They're not perfectly good at it, right? So they need a little help from their friends. And that's where these CD4 T cells come back in. And so CD4 cells don't directly recognize the virus and don't directly recognize the cancer. What they do is they have these antigen-presenting cells that will basically kind of like engulf a dead cancer cell and then present it to the CD4 cell and say, here's what's going on in the neighborhood, okay? And then the CD4 cell can recognize that, and what it'll do is it'll turn on its genetic program to help indirectly kill the virus or the cancer, right? And so it'll release molecules that will recruit more CD8 cells or activate the innate immunity to kind of help us clear it out or do things like that. And then it'll also tell the CD8 cell, keep going, you're doing a great job, okay? And so, these are typically thought about to be helper cells. What Marie in the lab has discovered is that she can now create CD4 cells that make the molecules that directly kill cells, right? They can make this granzyme and perforin. And so what I'm showing here is these, we don't even have a name for them yet. It's kind of that hot off the press, but we can make these cells, these CD427s, and you can see at 4 different experiments, we get 10 to 40% of cells that are making granzyme B, right? Whereas CD8s, where their professional job is to make granzyme B, you can see the 4 on the right here are much lower. So we're able to make these CD4 cells that can potentially directly kill problematic cells, right? And so how are we going to use this? What we're thinking is that what antigen-presenting cells would we want to eliminate from the body, especially when you think of autoimmune diseases, right? They present, food that you eat, that we get tolerized to that. They present debris that they find. But if we can engineer these cells to specifically recognize antigen-presenting cells that are presenting problematic antigens, so like gluten in celiac disease, if this antigen-presenting cell presents gluten, and we have an allergic reaction to gluten, we might want to destroy that APC, right? If it's an islet cell, so the cells that make insulin in the pancreas, right? These antigens are presented by these antigen-presenting cells, and our immune system mistakes it and then kills those cells. We could then eliminate the cells presenting these pancreatic antigens. And there's other ones. There's myelin proteins for multiple sclerosis, proteins in the brain, and then citrullinated peptides, which is actually another form of post-translational modifications. We could, effectively kill the cells presenting that and kind of dampen the immune response in a situation like rheumatoid arthritis where we don't want inflammation to happen. So what that means is that we are killing the messenger in this particular point, right? We want to get rid of those cells. So yes, so again, my lab studies the balance of the immune system in autoimmune diseases, where we again try to make technologies that we can apply and study things in a new way. So we can kind of connect this loop now, where we can do the scientific method, where we make a measurement or we make an observation, we make a prediction, and then we try to break that prediction or try to test that prediction by breaking the system, right? And we've created a loop now. And so what this allows us to do is we have these novel frameworks for looking at signaling transcription networks in immune cell decision making. We can study the signaling in immune cells to an unprecedented depth, right? Again, we're looking at 20,000 phosphorylation sites instead of 5 or 10. And then again, because we're doing interdisciplinary approaches, we have a multi-pronged plan of attack for a multi-pronged set of diseases. So with that, I want to say thank you. This is my lab. This is the funding, particularly GAI, and we have a website if anybody's interested. We have some fun stuff. So thank you so much for your attention. Okay, so typically we take questions at this point, but we're going to kind of do it a little bit more formally. So we're going to transition to the next part where Dr. Sapphire is going to join me on stage. And this is going to be moderated by Sandy Boak-Wernis. Sandy will also open the floor for questions from you and for the audience. So just give us a second while we set this up. Right on time. Joel, everybody. many are we doing? How many chairs? 3?
2. Sandy will stand right here.
Can
Yeah.
you introduce Sandy?
Do you want to do it from here?
Yeah.
Okay. Okay, so as LJI's first Global Autoimmune Institute professor here, it's my pleasure to introduce the leader whose influence has not just been visible in my work, but has kind of been woven into the fabric of LJI. Sandy's partnership between the GAI and our institute, a partnership that's been propelled by research that's equipped my lab with technology and people, has allowed us to reach new heights and established a key platform for scientific dialogue that I lead, the Walter and Jean Boak seminar series here at LJI. So to introduce Sandy is to appreciate the trajectory of a career dedicated to making an impact at every level. Her path began at Capitol Hill, where she wielded her legal expertise as assistant minority counsel to a U.S. House Select Committee, advocating for essential policy changes. The foundational experience led her to bring systematic leadership, or strategic leadership, to North Virginia Pathology, PC, where she merged her understanding of law and medical practice to navigate complex healthcare challenges. Today, as the founder and executive director of GAI, Sandy draws on all these experiences to fuel a mission that is both ambitious and profoundly personal, at least for me, while shaping the future of autoimmune disease and advocacy research. The Global Autoimmune Institute, under Sandy's leadership, is more than an organization. It's a force in rethinking how autoimmune diseases are addressed. Since 2015, GAI has championed cutting-edge work that extends from funding innovative research to cultivating comprehensive educational resources and fostering multidisciplinary approaches to healthcare. Sandy's vision stems from the deeply personal and all too common story of a misdiagnosis and dismissed symptoms as she advocated for loved ones. The drive that once fueled late nights on Capitol Hill and pivotal decisions in pathology now power a mission that insists on change in how patients are seen, how their stories are heard, and how science tries to rise to meet them. Sandy's leadership has not only shaped institutions, but has redefined how we view support. Her scaffolding, not just a safety net, as a dynamic partner, not just funding. It's a privilege to introduce someone who exemplifies what happens when knowledge and advocacy and an unyielding belief meet. So please join me in welcoming Sandy Wilk-Burness.
Thank you so much. That's wonderful. I'm so happy to be here and so happy to have the privilege and honor of being able to support and contribute to this incredible research. And that was a wonderful presentation. It was so very understandable, and I hope millions of people see that.
Thank you.
We'll do what we can to help that get disseminated. It's a privilege and a joy to be here today, both as a member of the LJI board and as the executive director of the Global Autoimmune Institute. I'm happy to be joined by my science-loving husband, Dr. Bruce Warnes, who serves as GAI's chief scientific advisor. We're thrilled to be a part of this event. I'm here today because of painful personal experience that brought home to me the extreme need of so many individuals who are suffering with autoimmune disease and concurrent disorders with very little hope for basic understanding, a diagnosis, for help and support, treatment, and for a cure. GAI represents the voice of the patient community. To them, we provide accurate information and comprehensive information, education, validation of the aspects of their experience and their journey, and most importantly, we seek to support research that will ultimately impact their lives. Research like that of Dr. Myers and his team and the other dedicated scientists here at LJI is the work, the investment that's going to save and improve the lives of millions. Now, let's get started. We would love to field some questions from the audience. And I guess I'm going to start. So, Dr. Myers, for those of us who don't work in a lab but live with autoimmune conditions, what aspect of your research do you think holds the most promise for altering our relationship with our health?
That's a great question. I think one lab can make a number of discoveries, and those can be impactful to a handful of people. But one of the things that I'm particularly proud of is these technologies that we built. Yes, we use them to make discoveries in autoimmunity, but other labs can use them too. So, we're not just one person doing it. We're trying to be more catalytic and make it so, other labs that are maybe even better at this than I am can go and try to make their own discoveries, and we get this propagating effect where more and more people can ask better and better questions about these diseases. So that's kind of what I think is going to be our biggest impact.
Well, that's fantastic. So it's collaboration and spreading the word. So
Yeah.
Dr. Myers, you've highlighted that immune responses are influenced by thousands of biochemical reactions happening simultaneously. Dr. Sapphire and Dr. Myers, how does LJI prioritize which pathways to study? And what surprises you most about when these priorities change in response to new data?
How do we prioritize them?
That's a question for both of you.
Probably in a variety of ways. I think one is if we find something and somebody else has seen it before, that's a lot of confidence that what we've discovered is we can trust it. But then on the flip side of that is if someone's never seen it before, that might be an interesting path, because again, we're doing these experiments and we're seeing things that people haven't looked into before. And so this might be a nice place to do it. So it's kind of how much we trust the result and where we think it might have the biggest impact. How do you choose?
I would agree with that. One of the things that LGI is just remarkably good at is gathering and harnessing the incredible complexity of data that we can measure on immune repertoire, immune history, all the little metabolites in cells from the foods that you eat, what sets us apart based on our genetics, our exposure, our history, the medicines that we take. And we've found these pathways that can harness this world of data and turn them into usable knowledge by figuring out where there is a strong signal above the noise. So one of these things is a database we call DICE, that's D-I-C-E, it's an acronym for the Database of Immune Cell Epigenetics. And using this database, our professor Pandurangan Vijayanan, or Vijay, can find genetic modifications that link to people that have a disease or do not have a disease, or things that are different between males and females, things that are different between severe disease and resolved disease. And when you find strong markers like that, that helps the rest of us go and look and see, using our tools, what those different pathways are. So we rely on our colleagues and a complementary suite of tools and different approaches to the problems to find something that is a major question in human health that science hadn't had the resolution to look at before, or the right tools, or the right combination of experts.
I would second that. I mean, I'm a chemist by training, and so I've been in immunology for less than a decade, but I'm surrounded by these people who have been doing it for several decades. So I can ask a maybe stupid question that they would have told me not to do, but I didn't ask first. And then we'll do something and we'll find something interesting. And then around then I have all these fantastic colleagues to say, this is the experiment you should do next. This is how this could be helpful. That second project I talked about has been that. We had this data, we didn't know how interesting it was, and we showed it to some colleagues and they're like, whew, you could do some things with that. So that would be another way we prioritize things, is through the knowledge of my colleagues.
That really helped set you all apart frankly.
Yeah, I think we have a question here if you want to save.
I'm going to make a two-pronged question, if I might, but might I go back specifically to your presentation? Because it is absolutely exciting to me that we're getting down to narrowing down at this point, and then being able to work with all of the data that you've collected, with amazing tools that you have to work with here as far as machines and everything, and the staff that you have, and the sharing of information. But I have two questions that are almost, now what's the next step questions? And that is, when you were looking at the last two conclusions you came to and where you were going to focus, the first one is you talked about turning up and down the CD8 T responses in order to address the disease, i.e. kill it.
Mm-hmm.
And I wanted to know, one, if you've come up with an idea of how to turn it up. You mentioned that there was a little lightning arrow early
Mm-hmm.
on, and I wondered if that lightning arrow meant something that you guys already have discovered, or is that part of your research? So that's one question. Second question is, how will you train your CDT427, are these cells, actual
They're cells,
cells, the cells, to focus on the places that you know that they're going to be more effective than your CD8 T cells for?
Those are great questions, and I don't even know if I have good answers for them. I mean, and not because we won't, just because we don't. And it's because it's so early in these projects. I would say with the tuning of gene expression, we're probably not going to make genetic mutations in people, right? I mean, that's a challenging thing to do, and it can, people are getting better at it, but we're not there yet. What I'm hoping is that this does is points us at signaling pathways, and maybe non-canonical, or like not the same pathways that we've always looked at, like maybe side branches. And there are so many small molecules out there that inhibit or can modulate these signaling proteins that we could potentially repurpose drugs that are already out there and safe. And my dream is that we could use maybe 3 or 4 of them in a really low dose so the toxicity's gone. But if you hit 4 parts of this pathway, you can turn that gene off without a sledgehammer affecting all the other. Like, we don't dampen the whole immune system with like corticosteroids, right? We dampen just the T cell response for this particular gene by 3 or 4 little small molecule tickles. So that's kind of where I'm hoping that goes. Training the CD427 cells, which now that I'm hearing it, that's a terrible name. We'll come up with a better name. That I don't know yet either. I think one thing is there's a process called CAR T cells where you can put in a receptor that recognizes a specific antigen. And so probably the first experiments we would do is make CAR T CD427 cells, right? And we'd pick the receptor for gluten or for a myelin protein, right? And those will have different kind of advantages and disadvantages to them. And so we're just gonna have to try things and see kind of where it sticks. So that's what I dream about these days. But yeah, I don't know if we have a good answer yet.
Thank you for your wonderful talk. I am just fascinated by the video. Could you share a little bit of the techniques you used to show us these microscopic interactions and activity, or were they just animations?
I downloaded them from the internet. No, that is, I tried to say it and I left the lab's name up there. So that's Alex Ritter's lab, and he does a lot of imaging, of live cell imaging, to kind of see how T cells interact with their target cells. And so what I imagine he did, I don't know that worked particularly well, but what I imagine he did is you can fuse proteins that give off a color, like a visible color. You can fuse those to a couple proteins that you're interested in the T cell or in the cancer cell, and then you can watch them move over time. So he might have had that granzyme B protein that I'm talking about, he might have had that tagged with a red fluorescent protein, and that's why it glowed red, and that's why you can see it move. Where he might have had a green protein tagging the outside of the cell. So, and then what you do is you incubate these cells together in the right temperature and conditions, and you just watch them with the microscope. I believe we actually have the capacities here.
Yeah.
I have no doubt that we could do something similar, but yeah, that wasn't my work. Those are just such killer images that I wanted to.
We've done that.
That would probably be 40x, right? Because you're looking at just 40x. You're looking at, is Z in the room? What do you think? So that's the microscopy expert. That's Z.
More about the details during the break, but we are doing those experiments for Dr. Sapphire as well.
We have this wonderful core facility for imaging, and we can average anything from an entire mouse all the way down to a hydrogen bond. With a suite of instruments, each one picks up where the other one left off. So you can see dynamic motions and actions of particles, and then once you get down to the molecular level, you can craft the molecule or craft the vaccine or craft the drug to take it all the way back up.
That's one of my biggest regrets about getting into mass spectrometry and proteomics. It's not very aesthetically pleasing science. So I have to steal images from other people and colleagues. I should have stolen it from her actually, but. I think it's recorded, so
Yeah, we're recording.
Hi there. I am great presentation. I'm not a scientist, but I love science, and I'll share a personal journey that I'm on an immunosuppressant that targets IL-17 A and F, and I wonder with the work that that's being done here specifically, or your feelings around the immunosuppressants, how much more targeting can be done to minimize the impacts on over-suppressing, let's say, of the immune system?
I mean, beautiful question. I actually think that drug is a good example of narrowing it down, right? 50 years ago, you maybe would have taken a corticosteroid that would have dampened your entire immune system. Now you're dampening a signal that is very specific to a subset of pathways, right? And so, it's not as bad as it used to be, but I bet it could get a lot better. So, you could imagine IL-17 is a signal that tells the cells to to turn on the inflammation pathway, right? But again, that's the signal that goes through the signal transduction. So there are potential possibilities where we could study that IL-17 signal transduction pathway and say, are there any vulnerabilities in there that we could take advantage of? So I would say, I think it's kind of a little bit of both. I think we have some places to go, but I mean, that's a big advancement from what it was, 20 years ago. So, not to minimize your.
Great. We have time for just one more question, but the rest of you, any other questions, please talk to people and talk to Dr. Myers and Dr. Sapphire during the reception.
I think we have one more, right?
Thank you very much for your presentation. What does LJI do about their discoveries to get it from the basic science to helping those of us who have autoimmune diseases?
I can take that. So we have a team led by Margaret Hing over there, our Chief Legal Officer, that looks for what is needed for the next step. Do we need clinical samples where we can partner with, for example, Moore's Cancer Center or other hospitals? We work with Cedars-Sinai, work with hospitals around the world. Or do we need more translational investment to move it forward? So I can give you one example of the work of Professor Mick Croft. So there's a molecule called OX40. It was discovered at Oxford, so they discovered that it exists. But what the molecule does was studied here by Mick Croft. And he realized it's a molecule that turns on different kinds of inflammation and modulates T cells. And so from his life's work of the basic research of understanding how these things work and how it controls the immune response, he reasoned that there was an opportunity for drugs or medicines that could dial that back down. And so working with our pharmaceutical partners upstairs that invest philanthropically in our research, Kyowa Kirin, we're able to together develop an antibody that is now the medicine rocatinlimab. We call it ROCA. And that's now in successful phase 3 clinical trials for atopic dermatitis with potential for more. And so sometimes we have built-in clinical partners and translational partners. Other times we look to other sites that have different kinds of manufacturing or translational expertise to move it forward. But it's an active thing that we want to do. We try the 1,000th experiment because we want to see our basic findings eventually become a medicine for people.
Thank you so very much. It's been a wonderful, wonderful presentation and session. And we're now going to hear a few more remarks from our wonderful and the great Dr. Sapphire.
I just wanted to thank all of you for joining us here today. Thank you for GAI for coming out from the East Coast to join us. Our involvement with you, our supporters, not only inspires us, but directly fuels our work, and we have you to thank for all of that. So please join us out in the atrium. You'll meet our young scientists, our Spark finalists that have posters. The heads of our mentorship program are here, and please linger, and we'd love to talk to you more. So thank you for coming.